Concepts in Molecular Biology for the Immunohematology lab. So molecular blood group typing, we can use it. Molecular blood group typing uses DNA-based methods to determine an individual's blood group genotype by analyzing the genes responsible for that blood group antigen expression. So some advantages over serological testing. It is useful when serological tests are inconclusive or impossible. So for example, in patients who have recently received multiple transfusions where mixed red blood cell populations could confuse traditional serology testing. Could detect blood group antigens at the genetic level. So it can identify blood group variants in silent alleles that may not express detectable antigens on the red cell surface. And we also have high sensitivity and specificity, so it can detect rare or weak antigen expressions which might be missed by serology. So clinical applications for this is we have for searching for multiple antigen negative units, so particularly beneficial in patients with alloantibodies where it is critical to find compatible donor blood lacking specific antigens. Complements traditional phenotyping. So while serological phenotyping shows the phenotype expressed on red cells, molecular typing can actually give a genetic profile which will allow for more comprehensive matching. We can also do prenatal testing and transfusion management. So molecular typing can predict fetal blood group status and can help in cases like hemolytic disease of the fetus and newborn. Some potential impacts from this is molecular typing could revolutionize blood banking by enabling precise matching and better inventory management, which could reduce alloimmunization risk and transfusion reactions. So applications of molecular blood group typing, we also have fetal DNA typing. So molecular methods can allow for the typing of fetal blood group antigens using DNA obtained from the maternal blood or amniotic fluid. This is especially important in pregnancies at risk for the hemolytic disease of the fetus and newborn, which can be caused by maternal alloantibodies against the fetal red blood cell antigens. So early detection can guide timely interventions and management. You can also use it for typing of donors for alloimmune patients. For patients who have developed antibodies against multiple red cell antigens, also known as alloimmunized, molecular typing of blood donors can help to identify compatible blood units that lack the targeted antigens. And this can minimize the risk of transfusion reactions and improves transfusion safety. Can also do screening and locating rare blood group phenotypes among blood donors. So molecular techniques enable large-scale screening to identify donors with rare or uncommon blood group phenotypes that may be difficult to detect by traditional serological methods. So maintaining a database of these type of donors can facilitate rapid availability of rare antigen negative blood when needed. You can also use it for determining the frequency of blood group polymorphisms in a given population. So molecular typing allows researchers in blood banks to analyze the distribution and prevalence of various blood group alleles and polymorphisms within different ethnic or geographic populations. This information helps in understanding population genetics and can aid in improving transfusion services by predicting the availability of certain phenotypes. Zygosity determination for fathers of fetuses at risk for hemolytic disease of the fetus and newborn. So determining whether a father is homozygous or heterozygous for a specific blood group antigen gene can help assess the risk of the fetus inheriting that antigen, which is actually critical in pregnancies where the mother has antibodies against that antigen. This can aid in better clinical management and counseling. You can also use it for blood group typing of patients with autoimmune hemolytic anemia and or other diseases. So in patients with conditions like autoimmune hemolytic anemia, serological blood typing can be complicated by the presence of autoantibodies that can interfere with antigen detection on red blood cells. So molecular typing can bypass these serological issues by directly analyzing DNA, providing accurate blood group information that is needed for transfusion decisions.